Hi, I'm Saurav, and thank you so much for inviting me to give a talk here. I'm a postdoctoral research associate at the British Antarctic Survey and associate postdoc at the University of Cambridge. And I work with Rowan Whittle at British Antarctic Survey, and we are trying to, as part of my project, reconstruct the climate of Antarctica across the Eocene-Oligocene transition. It's a mouthful, but I will explain to you what it is. Earth, as we know, like this is typical temperature curve by Zekos et al. And in the x-axis, you can see the temperature, which is marked in red, and the y-axis marks the age. So this particular graph is like the temperature diary of our Earth. As you can see, the Earth has fluctuated like temperature through time, and this record comes from the deep sea sediment records. The part which I'm interested is this region here, where you see a sudden decrease in temperature within a very short time interval. And that temperature decrease led to freezing of Antarctica. So the Antarctica, which we see today as this white lifeless desert, was actually not this 34 million years back. This is like a painterly representation of what was there. There were penguins of the size of 2 meters, which is like 6 feet, so taller than us. Uh, there were mammals, there were different birds, forests, and everything on the ground. And on the sea, we had sharks, crabs, and other predators. So this is one of the beak of these giant penguins which we found. It's almost a complete beak, and I think so far, like this is the largest or biggest beak we have found. And these are the shark teeth which are present in the fossil of Antarctica. Now, how do we know the temperature? How do we reconstruct temperature of somewhere which was so back in time? Well, the ocean kept records in the shells of the animals which lived in it. So this is a bivalve, um, and like it grows through accretion. So it grows from here to this to here, and it's almost like a tree ring where every growth ring is like a yearly development. Similarly, like they grow yearly or similar time frame, and so each growth band or growth line is kind of a postcard of the ocean on that day when it was alive. And I try to read the postcard essentially to try and understand what the conditions were. Um, Bivalves shells, they are made of calcium carbonate, which is the same material as chalk and marbles and stuff. But they take in this carbon and oxygen to build this calcium carbonate shells from the seawater which is surrounded. And accidentally or fortunately, they take in the chemical signature of the seawater as well in their shells. And we use this chemical signature. Bear with me, this is going to be short, but isotopes is what I use a lot for reconstructing temperature. And when I think of isotopes, I think of cats. So there is this thin cat and there is this fat or heavy cat. And isotopes are also same in terms of their different you know things of the same elements. So for oxygen, they belong like of the same, but the difference is only in mass. So think of it as the treats which we give to the cats. So if we give less treat, the mass is lower, and that's why it's called like 16 oxygen. And if we add more treats, which is adding more nutrients to the nucleus, the mass increases and it becomes heavier. So it's called 18 oxygen. So that's the fundamental thing. And isotopes, both the light and heavy isotopes, are present everywhere. So in the water where the shells live, in the shells as well. But the proportion of light is to heavy isotope in the shell depends, thankfully, on temperature. And it's like inversely proportional to temperature. So what it means is if we find more lighter as compared to heavier oxygen, the temperature is actually warmer as compared to when we find like more heavier to lighter isotopes. So since they are inversely proportional, that's how we find it. We'll get through it with examples further. But in order to get that shell, where do we find the shell and where do we find the fossil? End of warning. So Antarctica, as a lot of us know, is a desert of ice, but only 0.2% of it is ice-free, and Seymour Island is one of that place. And it's a place where we find a lot of fossils. This you see on the right is a geological map, which essentially showcases the beds. So the beds with fossils in them stacked over each other. So imagine this to be a chapter of a same book. And as we move from chapter 1 to chapter 8, the time actually passes. So this is the oldest and that's the youngest. And each chapter has fossils in it. So I collected fossils from all of these units. So 31 till 39, we have collected fossils from Seymour Island, and that's the island where we find samples from the time when Antarctica was still warm. And then the transition happened. And the the transitionary rocks are present in this island, which is a further north of Seymour Island, called the King George Island, where again you see here the geological map. And we went to each of these units and collected fossils from there. So this is supposed to be the time when Antarctica actually froze. So we have a bracketing time of Antarctica when it was warm and when it was cold in terms of uh, fossils being collected from Seymour Island, which is the warmer Eocene period, and King George Island on the right, which essentially showcases Antarctica when it froze. Today also, like if you look at the geology, it almost looks like Seymour Island is like full of mud and just rocks, whereas in King George Island, we still have glaciers and all. So yeah, that's somehow related, but yeah. And you can see here us, these are the campsites for us when we went there. So we stayed in those campsites and collected fossils. So my question for my project is to understand how cold did it actually get and how fast did the cold happen when the transition actually occurred. This is how Seymour Island is mostly full of mud. It's contrary to what you see in Antarctica. Yeah. So yeah, that was the challenge in Seymour Island, mud everywhere, inside, outside tent. Here is Sam, our field guide, trying to stitch the floor of the tent, which was not possible. The King George Island has a different challenge. The terrain was very difficult. It was a slope which went straight down to the sea, and there were glaciers as well on which we had to walk. So it was a different challenge. For some of the locations we couldn't go. So it's our again our field guide Andy. He's trying to see what's if there is any fossil in that part. So yeah. And there were different challenge. I was telling to Lydia like that Antarctica is supposed to be more desert-like, not rain, not much of rain. But when we went there, there was a period of a couple of weeks or just less when there was continuous rain and our like tents got flooded as a result of it. So yeah, different island, different challenges, but one thing in common which we try to find out are the fossils. So what you see on the left is a fossiliferous unit from Seymour, and what you see on the right is fossiliferous unit from King George. From the look of it only, you can see that the Seymour Island is almost like a pavement of fossils. There are many fossils out there, whereas the fossils in the King George Island are more like scattered. As a result of which, we try to collect the best fossils. So once we find a fossiliferous unit, we note down the geolocations and everything, and then I go first to collect my samples because they have to be really good looking samples for any geochemistry analysis. I'll tell you why. But yeah, that's the first step. So this is collected from Eocene. And then what we do, we bring them to the lab, we drill out powder samples and weigh them and run them in the machine. And the machine gives us the value. So as I told you, what you see, the values here is the proportion of uh, light is to heavy isotope. So what you see on top is actually more lighter as compared to what is in the bottom, which is heavier. And as I told you, it's irreverse. So it's not directly proportional. It's indirectly proportional. Hence, if you do the temperature conversion, the one with the lighter isotope should be warmer as compared to the heavier isotope. Thankfully, we have established equations for doing this. And once we put our isotope values in the equation, we get the temperature. And as I told you, like the lighter isotopes have like higher temperature as compared to the heavier isotopes. So, but it's it's a story which goes well with shells which you collect from the ocean right now. Remember, the shells are sitting on the rocks for more than 34 million years. And that's why we have to check for any sort of uh, change in chemistry which has happened after the shell has died and since the time, like from the time we took it. So that phase of time after burial till we receive it, the shell can go through a process known as diagenesis, which can completely change the chemistry of the shell. And that's why this is one of the reasons why I look out for the best shells, which are best or good looking shells. But that's not enough. We have to go through much more detailed screening process. One of the easiest screening process is to compare our data with the previous published data. So that's what we did for the Eocene because a lot of work has already been done on the Eocene samples where you find like a lot of fossils. Irrespective of the method they used to find the study, here we see the temperatures they reconstructed, and we go from old to the young formation. And as we move from the old to the young, we always see a temperature decrease. So it's moving from a warmer temperature to a colder temperature. Mostly the trend is that. And the most accepted study so far is by Ivani et al., where she showed that the temperature actually moved from 15 degrees Celsius to 5 degrees Celsius. So it's a decreasing trend of temperature just before the transition can take place. And so this is her data. Uh, so what we see here is the isotope values. This is oxygen isotope. Similarly, we have carbon isotope, which is an independent isotope, and it tells us a different story altogether. But yeah, we get both of them together. And on the y-axis, we have age. So moving from older to younger, we do see a decreasing trend overall. But then we have our own data from the same place, from these two bivalves. So the red one is this particular bivalves, which is now called Retrotapes, which is this. And the blue is the Cuculia bivalve, which is this. And when we put our data on top of it, we can see there is no such difference. So we can clearly say that yes, our data is showcasing kind of the temperature when the shells actually lived. So we call it preserving the pristine characteristic of the shell. But when we and like based on that we plotted our curve, and what you see here is time. So these are like the chapters of the book I mentioned. So from 31 to 39 we are moving from old to new, and we see that the temperature actually decreasing. And from here you can see it's almost 15 degrees Celsius to 5 degrees Celsius, which goes well with the study so far. But King George Island has been least studied. Like it has not been studied much. These are the couple of studies based on which we went and we found our field locations and we collected fossils from. And one of the study actually mentions to be cautious about using shells to reconstruct temperature because they can be altered. And if you look at the condition of the shells, you can decide. But this is a very important place for fossils. As you can see here, we found really unique fossils. This is an echinoid, and what you see here is a ophiuroid. Now, when we, or like brittle star, uh, when we see the modern Antarctic seafloor, so this is modern Antarctic seafloor, we see they're filled with these ophiuroids all around. And from the King George Island, nobody has discovered the ophiuroids so far. So we were like one of the first who actually found evidence of ophiuroids. So we think that they could be the most recent ancestor from the present seabed creatures. But yeah, we have to do more study to actually say that. But this makes it important why the shells from King George Island are so important. However, they look like this. One of my collaborator actually labeled them as shitty shells because they are present like this, they are stacked together. It's very difficult to get them out. And when we find one, we try to take it out and it just explodes. It just breaks into multiple pieces. So I had to adapt a completely different method to collect this specimen. So in field itself, when they break apart, I collect them separately and put it in one bag so that they're representative of one individual at least. This is one of the rarest shells which got back to the lab safely. So this is like the precious shell which we have, just the one shell. It survived all these atrocities. Now these are shells which has not been studied before. So what do? We don't have anything to compare our shells with. So we need to come up with a screening process to check if they have preserved the original signature from the time when they lived. And for that, the first screening tool we use is called scanning electron microscope. Just for context, like our regular hand uh, glass or light microscope can go up till like 400x or 1000x times zooms. But with this technology, we can actually get like 1000 times zoomed in like photo, or we can like zoom in so much in depth so that we can see the microstructure in it. So using this method, we compare a recent shell from Antarctica as from like the fossil shells which we collected. And when you look at the external view, this is how they look like. So this is the recent one, and this is a fossil one. So the pattern which you see, they're very much like uh, similar in terms of that there is no blocky, patchy, uh, surface which is coming out. So that kind of showcases that, yes, the preservation has been good for our fossils. So this is one view, and then we see it from the posterior view. And when we see that, that's how the recent looks like, and that's how the fossils looks like. Again, we see the patterns are repeating nicely, and there is like foliations within the shells. So there is no patchy arrangement to it, which kind of confirms that, yes, from the posterior side also they look good. And then we do a side view where we cut out the shell and try to find out what's inside. And this is how they look like for recent shells. You can see this foliation. This is a very thin shell, these two. So, but you can see the foliations, like the bands stacked together, which kind of confirms that they are preserved well visually. But then the next screening process happens at a chemistry level. So this is energy dispersive X-ray spectroscopy, also known as EDX, which is in the ACM. And what it does is it releases electrons on whatever surface we are trying to look at. And once it hits the surface, they release X-ray. And depending on the element, the signature of that X-ray has a unique signature. So we can actually tell exactly at what spot what element is present, essentially. But it's not very precise, but it's very useful. For this, we try to look at some areas in the shell. For example, this dots which you see here, these black dots are actually created by microorganisms. So these are called microborings. So when the shell is alive, they don't do it. But when the shell is dead and it falls on the surface of the sediment, these microborers, which can be forams, uh, some worms, they attach to the shells and they start like creating this space within the shells. And then the shells gets buried and gets engaged with the water which is in the burial sediments, and they interact with them. And if at all, these pore spaces are where those elements gets deposited. And this is like a good signature for it. And when we see them with the EDX, we find like distinct glowy features here, which are manganese. And here you can see these white spots, they are actually the microborings. So this suggests that the water which the shell went through after it got deposited in the sediment was filled in manganese. But do we find it over like everywhere? That's what we try to find. So we compare that space with somewhere where there is no microborings. And when we do that, we don't find specific patterns except for some like singular points where they are stacked up. So it's a good news. We know that where there is no microborings, we don't find this diagenetic fluid effect. Now, what happens when we look at like a breakage surface, which is just exposed? So essentially the top layer is gone and now it's just exposed. That's where we find the most pristine signature. Like we don't find any kind of diagenetic fluid encountering the space. So that gives us the idea that if we just clean the upper layer and collect samples from the exposed part, that's the part where we get the best signature or the most pristine signature of the shells. So keeping this in mind, we went to do further analysis. Remember, we collected powder, so they can be run in machine and we can get a precise measurement of the elements. Now, this trace elements is essentially, it means parts per million or less, which if you, for reference, like we try to find out how much salt, like a pinch of salt, is present in a whole swimming pool. So modern instrument can give us that resolution to actually figure out how much element in that trace amount is present. So in terms of shells, we do it. And as I told you, manganese and iron is what I'm going to use as my case study elements, because when the shell is alive and actually moving, the seawater around is very oxygenated, and that means that the manganese and iron are in a insoluble state. So you can imagine that as them being staying in a solid phase. So when the shells are there and they're in the solid phase, they don't interact. Hence, like if we find a pristine shell, then it will have a very low manganese and very low iron. But when the shell dies and goes, like decays and goes under the soil, that's where the scarcity of oxygen happens. And that's where manganese and iron, they turn into this soluble form. So essentially, you can think of the solid forming into a liquid form. And when the shell interacts with that's the time when they can decay the shell and actually get inside the shells, and hence changing the original signature of the shell. So ideally, like an altered shell would have a elevated manganese and iron in it. Now, from our case study, this is what we find. For reference, these are our like modern day shells. So these are recent Antarctic shells, and as compared to them, these are our fossil shells. So they lie somewhere at zero. But these fossils, they are going like most of them are above 1,000. So they are elevated in terms of manganese, which suggests that the manganese has actually altered the shell. So if we just go with this plot, it says that we cannot use our shells for reconstructing the temperature because this has been diagenetically altered. That's what it says, like it's roughly 32,000 times higher. Now, yeah, given just this graph, we will exclude all our data. So all my field trip, everything goes into vain. But remember, we have iron as well. When we look at the iron, it's very interesting that when we compare the recent with the fossil ones, there is a huge overlap, with some exceptions, which are like really high values, which are clearly outliers because they have been affected by iron as well. Now, based on this study, it gives us hope that, okay, the iron couldn't seep in and change the shell. So, what does it mean in terms of the preservation? It essentially means that the manganese, when there is less oxygen, they get into the dissolved state faster than iron. So, we caught our shell somewhere in between here where the manganese has already affected the shells, but the iron couldn't. And the isotopes which we are looking at, they're far behind. So we can tell that the shells which are based here are actually our pristine shells. So we can actually use them to reconstruct our temperature. And that's what I eventually did. So getting rid of everything outside our expected thing, we got this value where you can essentially say that they are pristine. And based on that, we could reconstruct the temperature of the King George Island as well. So this is Seymour and this is King George Island. So we already saw decreasing temperature from Seymour, like within Seymour Island, but we were expecting Antarctica to be already frozen during this time. But it seems like the temperature stayed constant. Now, this is an ongoing research, and we are still trying to figure out what's happening here in this section. Why the temperature is still comparable and almost similar to the Eocene part. But as you can see, like when the transition happened, there was a steep decrease in temperature. But in our case, it's not. So that's something which we'll try to figure out further. So in conclusion, the Seymour Island samples, we see that there was a temperature decrease, and that's confirmed by our oxygen isotope analysis. But in King George Island, the overall cooling kind of paused. And so for future work, the first thing which I'll do is something called sclerochronology. So as I told you, the shells preserve almost every year on their shells. So if we collect them very finely, we can actually tell about the seasonal temperature. And we also need to find the age of those specimens. So we can do that using a different isotope system, which we plan to do. And that's part of my future work. And thank you all for listening. This is our group from Seymour Island and from King George Island. And yeah, this is a photograph. Like one of my collaborator always says, if you don't show your best penguin photo and you are an Antarctic researcher, it it doesn't count. So